Today we are going to discuss the technique PCR. PCR is polymerase chain reaction. This technique is used to amplify a specific region of DNA. For example, if this is a DNA sequence and if we want to specify a specific region of the sequence, we can amplify it by using this technique known as PCR. PCR is polymerase chain reaction. Once again, I'll tell you, it is used to amplify the specific DNA sequence, and this technique was invented by Kary Mullis in 1985. Kary Mullis in 1985. Principle of this technique is that the particular DNA molecule is heated up to a high temperature, and then these two DNA strands gets separated, give rise to a single stranded molecule. For example, this is a double stranded DNA. At high temperature, we can separate or denature these two strands, and after that primers will anneal to it, and further extension of DNA strand will takes place. One by one we will discuss all these steps in detail. Extension of primer will take place. One by one we will study all these steps in detail. Now come to the requirements for PCR. First of all, we need DNA sequence, the DNA which is to be amplified. So DNA template is required. This is the first requirement, DNA template. After that we need primers. What are primers? Now, primers are oligonucleotides, usually 10 to 18 nucleotide long, which hybridize to the target DNA region, one to each strand of DNA. For example, there are two strands of DNA, so two primers are required. One will anneal to each strand of DNA. So two primers of such a sequence are required. Then we need DNA polymerase enzyme. In PCR, a specific DNA polymerase, a special DNA polymerase enzyme is required. For example, Taq polymerase. Taq polymerase is the polymerase enzyme which have been isolated from a thermostable bacterial species. Taq is Thermus aquaticus, Taq polymerase. This enzyme was isolated from the bacteria Thermus aquaticus. And this enzyme, this Taq polymerase enzyme, is stable at temperature above 80 degree centigrade because Thermus aquaticus is a thermostable bacteria. After that, we require DNTPs, that is DATP, DGTP, DCTP and DTTP. We also need buffer. And after all these requirements, the machine which is used for PCR is known as thermocycler. This is the machine which is required for PCR. Now, I'll explain this PCR, reaction of PCR by this particular animation. Yes, see, this is PCR technique used to make multiple copies of a DNA segment, as we have already discussed. And we can detect pathogenic virus, bacteria, we can identify individuals' DNA by this technique. And the requirements are the sample, DNA we can isolate from the sample, then this PCR machine. After that we can get copies of the target DNA. Now, this is thermocycler. As I have already told you that PCR technique is carried out in a thermal cycler. This is thermocycler machine. Now, and this is Eppendorf tube in which we will add DNA template, primers, nucleotides, that is DNTPs, and Taq polymerase enzyme. After adding all these things, we'll keep this Eppendorf in our PCR machine, that is thermocycler, and then set the temperature and number of cycles in this machine. Now you can see that this is a double-stranded DNA and this particular sequence is the target sequence. Now there are three cycles of PCR. First is denaturation, which carries at 95 degrees centigrade. At this particular cycle, the hydrogen bonds between these two segments, these two strands, gets broken. This is known as denaturation. Now these two strands are separated. Then second step is annealing. See, these are the primers in the second step, which is in which temperature lowers down at about 55 to 65 degrees centigrade. Primers attached to their specific sequence. Third step, which is extension. Here these are the polymerase enzymes which starts extending the DNA sequence from the primers. Primers are attached at the 3' end, and see, these polymerase enzymes carry the process of extension. Again, this is cycle 2. In the second cycle, again the same process applied, and from the 2 strands of DNA, now we'll get 4 different strands of the same DNA. Again, denaturation, annealing and extension takes place, and see, you will get 4 different DNA. We started from the 1 DNA segment, 2 strands of DNA, and after that 4. Means they will increase with they will increase at the rate of 2 raised to the power n, where n is the number of cycles. See, this is the third cycle. Again the same steps are repeating one by one, and we'll get multiple copies of the DNA. At the end of all these cycles, you will see that you have many copies of the DNA required. At the end of cycle 3, target DNA copies are 8. 2 raised to the power n means 2 raised to the power 3, that is 8. After cycle 4, again 2 raised to the power 4. Like this, the number copies of your target DNA gets increased, and you will get multiple copies of the DNA which was required. Now you can use this DNA for various purposes.